Hi, everyone. Thank you all for being here today. It's my pleasure to be here and share with you some of my new analysis that come out of a spin-off project of Speed for Science. But before I start to introduce the spin-off project and talk about the analysis, I wanted to start with some background information about some of the things that we know and some of the things that I think we don't know, or we know only very little about substance use disorders. So what do we know? First, I think we know that alcohol and substance use problems are very prevalent and common in the United States and across the world. So a recent national estimates based on adults who are older than 18 years old in the United States indicates that about 14% of them had an alcohol use disorder in the past year, and about 30% have had an alcohol use disorder in their lifetime. So we're talking about a very prevalent and significant public health concern. We also know that alcohol and substance use problems tend to increase from adolescence and then peak during young adulthood, meaning around the age of early or mid-20s, and then decline afterwards. So this figure here shows a study that used data from the National Longitudinal Study of Adolescents and Adults. It basically shows how the level of heavy drinking change from early adolescence to mid-30s. So we can see that there's a peak there at around mid-20s. And this pattern is basically consistently observed for a variety of alcohol and substance use outcomes, so including frequency of drinking, alcohol use disorder problems, and smoking, marijuana, and a wide range of outcomes here. Although I wanted to note that this pattern that I'm showing here is the average. So people, individuals can look very different in terms of their trajectories of alcohol and substance use over time. So some people may be consistently higher in terms of alcohol problems, whereas others may be high in adolescence and then very low in adulthood. So we also know that genes are important. We know that our genes play an important role in how frequent we drink, how much we drink, and the likelihood of us developing alcohol and related problems. So just step back a little bit to talk about how do we know that something can be genetically influenced. So this would be a really brief behavioral genetics 101 here. So a common method that researchers use to study genetic influence on a behavior or a trait is to make use of the twins who kind of provides us a natural experiment to help us to understand genetic influences. So the idea is that we have monozygotic twins or identical twins and dizygotic twins or fraternal twins. So for monozygotic twins, the assumption is that they share 100% of their genes and 100% of their home environment if they are raised in the same house. Versus for dizygotic twins, they share 50% roughly of their genetic variation and also 100% of their home environment if they are raised together. So the idea is that if we look at how similar are these twins in terms of their behaviors, in this case, for example, drinking, and if we see that the similarity in terms of behaviors are higher for monozygotic twins than for dizygotic twins, then there's an indication that, okay, maybe genes play a role here because the twins who share more of the genetic variations look more similar in terms of their drinking behavior. Versus if we see that while MZ twins and DZ twins, they look about the same, similar in terms of their behavior, then it tells us that maybe genes don't matter. It's all about the home environment that they share, the common environmental experiences that they share among the twins. In another scenario is that if we see that monozygotic twins who share 100% of their genes and 100% of their home environment don't look exactly the same, that means maybe there's something else that's going on. Maybe there's unique environments that's not shared between these twins that also contribute to their drinking behavior, for example. So this is a very basic, brief idea of the twin studies. We also call ACE model. So what we have is really, what we are doing here is really partitioning the variance in a certain phenotype or a behavior, drinking, for example, to three components. A, additive genetic effects, the contributions of our genes. C, the common environments that share between the twins in the home environment. And E, the unique environmental experiences that makes the twins different. So the concept of heritability is really about the proportion of the variance in our behavior or phenotype that's accounted for by our genes. So if you divided A by P here in this formula, it gives you an estimate of the heritability for a certain phenotype. So here is a table that shows estimates of a variety of traits, and just give you an idea about what the heritability for alcohol and drug dependence and related problems look like in comparison to other traits. So on average, there's a whole lot of studies that has been conducted to estimate the heritability of alcohol problems and related outcomes. And of course, there are some sort of variations in terms of the results that they reach at. But overall, it's thought that about 40 to 70% of the variance in alcohol and drug dependence are due to the genes. Note that there are some differences in terms of the estimate of heritability as a function of the specific outcomes that we look at. So for example, if we look at alcohol consumption versus alcohol dependence, we tend to see that the estimate of heritability is greater for alcohol dependence, which we think represents later stages of the problem. And also there are some variations across developmental age as well. So we tend to see a relatively higher estimate of heritability. So our genes seem to become more important in influencing our alcohol problems as we get older. Another important thing that we know about substance use disorder is that not just that our genes are important, but how important our genes are is in part dependent on our environmental experiences. Yeah, these also gets at the notion we usually call gene by environment interaction effects that I will, in the next couple slides, including this one, shows a couple examples. This one is a study by Dr. Danielle Dick, used data from the Finnish twin studies. So basically here, the different color lines shows the estimates of the AC and E that we just talked about. So the blue line shows represents the effect of our genes. So basically what this figure presents is that as the level of parental monitoring increase, the influence of our genes, additive genetic effects, decrease for substance use behaviors among adolescents. Versus if you look at the left side, when the parental monitoring was relatively low, we had a much larger effect of genetic genes on substance use behaviors. So this kind of gets at the idea of opportunity versus control. So the idea here is really that in environments where there's more opportunity for our genes to express, to influence our substance use behavior in this case, you would be able to see, you would tend to see a stronger genetic effect versus in cases where there's greater social control that limits your genetic influences on substance use behaviors. Here's just another example for gene by environment interaction effects. This is based on a study recently published by one of our postdocs, Dr. Peter Barr in the lab. So here it shows is that social support is an important environmental factor or experience that can modify our genetic influences on heavy drinking for young adults. Although it's interesting here that it shows opposite direction, opposite patterns of interaction effects for males and females. So for example, if you look at the left-hand side, the solid black line shows represents the genetic effects. So for females, higher social support, as social support goes up, the genetic influence increase, so our genes become more important in influencing heavy drinking. Versus for males, we see an opposite pattern of the effect. So I think it's really interesting, but I'm gonna stop here and not go into the details why. We can talk more afterwards or come ask Dr. Peter Barr. So there's really a lot of evidence out there that genes and environments interact to influence substance use behaviors. So here are just a summary of some of the key important ones. For example, we found that genetic influences are more important in environments where parental monitoring is low or when peer deviance high, so when a lot of your peers also engage in substance use behaviors. And also our neighborhoods matter too. So in neighborhoods where there are more people move in and out, there are more alcohol outlets, making access to alcohol more easy, or living in an urban versus rural neighborhood, that can increase the genetic influence on our alcohol use behaviors too. And also in more stressful environments, for example, people who experience more negative life events, people who experience childhood maltreatment, we tend to observe a stronger genetic influences on their substance use behaviors. The examples that I showed earlier are all based on twin studies, so it's based on we call latent genes, latent genetic effects. So basically, we infer genetic effects based on the observed relationships between in terms of behaviors between the twins. But I just wanted to note that these patterns of gene by environment interaction effects are also observed in studies that actually measure specific genotypes. So we are seeing similar patterns of results across different type of genetic studies. I think another important thing that we also know is that the pathways of risk that contributes to our alcohol and substance use problems may differ across populations. For example, may be different between people of different racial ethnic backgrounds. So for example, we know that compared to European Americans, African Americans on average report lower rates of alcohol and substance use. However, we also know that on average, they tend to have more negative social and health consequences because of alcohol and substance use. So for example, we know that African Americans tend to, or more likely to have legal problems or police encounters after drinking. We also know that African Americans with alcohol and substance use problems, they are less likely to get treatment and care, and so they are more likely to have long-term health consequences compared to European Americans. Another thing is that we also know that a lot of the factors that we just talked about, like parental monitoring, peer deviance, peer influence, they're all important across populations. They're important for African Americans and European Americans. However, the effect of these factors tend to be weaker for African Americans. So they're not as salient of a risk or protective factors for African Americans in general. So maybe there are other factors that we need to take into account for people of specific racial or ethnic minority backgrounds. In addition, when we talk about genes, genetically we know that because of difference in terms of ancestry, there are racial differences in, there are differences in terms of allele frequencies and then the patterns of linkage disequilibrium across people of different racial ethnic backgrounds. So these differences could potentially also affect our understanding or test of genetic effects. So it would be important to take into account these underlying differences in terms of both genetic and the environmental level to understand, better understand pathways of risk to these alcohol and substance use problems across different groups. So a little bit more about racial ethnic minority populations. Because of their unique background, they also tend to have unique experiences that majority populations don't experience as much. For example, ethnic minority populations tend to experience more stressors, for example, including racial ethnic discrimination, microaggressions. They also, on average, tend to have lower socioeconomic status, also more likely to live in disadvantaged neighborhoods. All these could be risk factors that contribute to their alcohol and substance use problems. On the other hand, the positive side, racial ethnic minority populations also have their unique protective and resiliency experiences. For example, in a lot of racial ethnic minority families, cultural socialization is a common practice. So parents would oftentimes engage in this practice of teaching their kids about what their race ethnicity means to try to promote their sense of pride for their ethnicity or race. That could serve as a protective factor for their kids' development. And also a sense of racial ethnic identity, so the sense of my race, my identity is important to me, I love being me, I love my race, these ideas could be protective as well, and has been associated with positive psychosocial functioning in ethnic minority populations. So we've known a lot about substance use problems. However, I do think there's a lot of things that we don't know too, and here's one. I think that because a lot of the studies on how our genes interact with our environmental experience contribute to alcohol and substance use has been focused on populations of European descent. So there's really a lack of understanding how these processes may apply to people of other racial ethnic backgrounds. We cannot, because the differences that we just mentioned, we cannot simply assume that they can be generalizable. So there's really a need for this kind of research that specifically focus on ethnic minority populations. And also, there's really a lack of studies that look at experiences that are unique to ethnic minority populations to look at how those risk or protective processes may modify genetic influences on alcohol and substance use problems as well. So with that background, here leads to these cultural experience and alcohol use study, which is a spin-off project based on Speed for Science. So for those of you who don't know, Speed for Science is a large longitudinal study happened here at VCU that collects several cohorts of VCU freshman students and follow them up throughout college years and after they graduate to study how genetic and environmental factors come together to influence their behavioral and emotional health outcomes. So there's really rich data, genetic and environmental data in Speed for Science. So the goal of this spin-off project is really to capitalize on that rich data available, and then we recruit a subset of the participants in the Speed for Science study to specifically those self-identify as African American or Black. So we recruit them and have them complete a set of, we think, pretty nuanced measures that measure their race-related experiences, which were not available in the broader Speed for Science project. So with that, our goals here is just look at how those race-related factors, both risk factors and protective factors, may affect alcohol and substance use problems in the sample of African American young adults. And second, we want to look at whether there would be gene by environment interaction effects, specifically with these race-related factors in linking to alcohol and substance use outcomes. So in the next few slides, I would describe some of the key measures that we put in the spin-off study. So the first one we put in is racial discrimination. So basically, this is a measure that includes questions that ask whether the participants feel like they have perceived that they have been treated unfairly because of their race by different people in different occasions. And we ask them about how frequent that happens in the past year, in the lifetime, and how stressful that they felt because of that experience. We also ask about racial microaggressions. So these taps at the more subtle statements or behaviors that also make people feel like they are discriminated or because of their race. So this is a multidimensional measure that gets at different dimensions, for example, about assumptions that they're not good, not as good as other people because of their race, assumption of criminality, and also micro-invalidation, assumption that they're similar with other people, all other people of the same racial background. and also some microaggressions in workplace and school. We also measure racial socialization by parent and by friends. So this is also a multidimensional scale. So it taps at 3 categories of socialization practices, including, for example, behaviors related to cultural socialization. So these are behaviors that aim at promoting racial and ethnic pride. And also behaviors that taps at the idea of preparation for bias. So really emphasize on, because of being Black, I may be more likely to experience racial bias and how to cope with that. And also behaviors that may promote mistrust to people in the majority group. We also ask about racial identity. So this is also a multidimensional scale that look at different dimensions of this construct. So including centrality, basically how important as my race, ethnicity is being Black means to me. And private regard, so how do I feel about being Black? How do I feel about my race? Public regard, how other people feel about Black people, how other people respect or not respect people in my race. And of course, alcohol problems, which is one of the key outcomes that we study, we focus on in this project. So alcohol problems in these studies measured by the alcohol use disorders identification test. So it's a 10-item screening measure that assess alcohol problems. So here's the sample. We started with recruiting participants from the first 3 cohorts because people in the first 3 cohorts, they already have their genetic data genotype and available to analyze. But we only got about 150 people from that cohort. So we expanded to invite participants from other cohorts to participate as well. So that's why you're seeing different ends here. So we have 426 participants who completed the survey that we sent out. And among those, we have 154 who complete the survey and also have genotypic data ready for us to run some of the gene by environment interaction analysis. We can see that we have more female, so it's a predominantly female sample. Here just shows some of the descriptive statistics across for the key variables. Just some interesting things to know. The highlighted ones shows significant gender differences. So I just think it's interesting that on average, people indicate having experienced more than 17 out of 38 a scenario of microaggressions over the past 6 months. And it's also interesting that females reported experiencing more racial discrimination, and then parents seem to engage in more of the promotion of mistrust practice with males. And then on average, the participants endure pretty high level of centrality and private regard in terms of their race, ethnicity. And we see that those scores are relatively slightly higher for females than for males. So getting at our aim 1, to look at how these factors are related. So this table presents some of the correlations. Here, these numbers shows how the race-related factors are associated with alcohol problems. We can see that experience of discrimination and microaggressions are associated with higher alcohol problems. And then the racial socialization and ethnic identity also are correlated, but only for some of the dimensions. And we can also see that overall, these race-related risk and protective factors are generally correlated with each other with some exceptions. And the correlations are modest, so indicating that they are related but unique constructs to look at. So we move on to ask this question, really, does racial socialization, does learning about your race, could moderate the effect of experiencing discrimination or microaggression in terms of linking to alcohol problems? So here, these tables summarize the results from analysis predicting alcohol use problems from two models, one model looking at the moderating effect of parental socialization and another looking at peer socialization. We see that for both models, racial discrimination is associated with higher problems, higher alcohol problems. But then in terms of main effects, there's no main effect of the racial socialization by both parents and peers in terms of linking to alcohol problems. However, we see significant interaction effects, so indicating that peers' racial socialization, so specifically cultural socialization practiced by peers, kind of buffers the negative effect of experiencing discrimination and microaggression on alcohol problems. Whereas the preparation for bias and promoting mistrust exaggerated the negative influence of these discrimination and microaggression experiences. Here we run the similar analysis, but look at microaggression. And we also see a significant interaction effect between peers' promotion for mistrust and experience of microaggression. So these results suggest that peer promotion of mistrust increase the negative influence of experiencing racial microaggression on alcohol problems. So we also look at whether racial identity can buffer some of these negative effects. So here on the left-hand side is the model looking at whether racial identity buffer the effect of experiencing racial discrimination, and on the right hand is for microaggression. And we can see that in terms of main effects, there's also no significant effect, protective effect of racial identity on alcohol problems. However, there's this interesting, I think, interaction effect that indicates that private regard, so I love being black, I feel good about my race, really buffers the negative effect of microaggression on alcohol problems. Moving on to aim 2. So here we look at whether these race-related experiences or risk and protective factors may interact with genetic predispositions to influence alcohol use problems. So just a couple of notes about measuring genetic risk. Here we actually have the DNA. We have our genotypes. So we know that many genes contribute to alcohol problems, and so it's polygenic. It's influenced by many, many genes with small effects. So nowadays, a common approach to measure our genetic risk or genetic predispositions for a certain trait is to use what we call genome-wide polygenic scores. So basically what it does is to use estimates from a large, a genome-wide association studies for alcohol, for example, that use very large discovery sample and calculate a polygenic scores that represents aggregated genetic risk across the genome for that particular trait. So large discovery sample size is really important. So for our particular analysis, we use one of the largest GWAS study for alcohol consumption available to date, which is based on the UK Biobank sample, to calculate a polygenic risk scores for alcohol consumption. So here are a summary of the results that we see, that we find. So we calculate polygenic scores across a series of p-value thresholds, and then here, these columns shows main effects for the genetic risk. And then here is the genetic risk by discrimination interaction effect. And then here is the genetic risk by microaggression interaction effects. So you can see across the board, we don't have a statistically significant effect, genetic or G/E effects here, as indicated by the p-values all greater than the conventional threshold 0.05. But I do want to point out that we kind of get some consistent estimates for genetic risk by microaggression interaction effects here at a reasonable effect size, and then the p-values are getting close to, getting more close to being significant. But overall, so far, no evidence of gene by environment interaction effects here. Here are some of the reasons that I think is making these happen, like leading to these non-significant findings. First, our genetic analysis is really conducted with a very small sample size, particularly in terms of genetic studies. We have less than 160 individuals in this set of analysis. So we overall has pretty low power for our analysis. And second, I think that currently our measurement of genetic risk for alcohol outcomes are not doing a very good job. So although we have a pretty large discovery sample size, but still the polygenic scores for alcohol outcomes are still account for a very, very small amount of variance in alcohol outcomes. So we are talking about less than 2% mostly. So we are not doing a great job measuring these risks. And this issue becomes more, not problematic, not more significant in the context where we don't have matching ancestry in our discovery sample and our target sample. So the genome-wide association study was based on European American samples primarily, and we are focusing on African American sample here. So probably this mismatch is creating more issues in terms of prediction accuracy in our polygenic risk scores here. Or maybe there's really no effects there, although I remain question about that possibility. So to summarize, there is some evidence that experience of discrimination and microaggression because of one's race and racial socializations and ethnic identity, all these race-related factors play a role in alcohol and substance use problems in African American young adults. And then there's also some evidence that racial socialization and identity may moderate the effect of experiencing discrimination and microaggression on alcohol problems. And so far, there's no evidence of gene by environment effect, although we'll continue to explore these questions, and we hope to find new things. So here are a couple next steps I'm thinking, and also here's where I welcome your input. So I'm thinking in terms of next steps, I could expand the analysis to look at other polygenic scores, for example, for education and for depressive symptoms, where large discovery GWAS has also been done. So we also have like kind of better predictive polygenic scores to see how polygenic risk for those other outcomes may interact with race-related outcomes factors to predict alcohol problems. And we could also extend to other outcomes, including cannabis use, depression, anxiety. We could also test for potential gender differences or run within gender analysis to explore what happens. So with that, I just want to acknowledge that the spin-off project is in part funded by the VCU Postdoctoral Association Small Research Grant. And I wanted to thank all the participants for the project, and Dr. Danielle Dick, and all the Edge Lab members, and also my collaborators from psychology and other departments. Thank you.